here in the view, filling in for Rich Reddy, who unfortunately is not able to be here today, but I'm pretty excited because Raina has a fascinating work history I'm really excited to hear about. Want to hear your rough cuts. But before I introduce Raina, I wanted two announcements. One, please join us this Friday. The IOE is having its monthly first Friday at Bridger Brewing from 5 to 7. Bring yourself, family, friends are welcome. Bring your grad students, bring your advisors. It would be great to have as many people as possible. It's actually always really fun opportunity to just have a beer and visit with each other in a very relaxed and casual setting. So please come. And then I also just feel compelled because as long as I've been doing these rough cuts last fall this spring, no one has done this before, but I want to introduce all of you also to Amelia, who is our wonderful tech here. And she's so awesome, and no one's ever given her a shout out. She's so dependable. She's here every week. She always gets the technology figured out, knock on wood, and she's just been fantastic. I just want to use the credit. So, Raina Plowright is here today to give her rough cut talk. She's an assistant professor here at NSU in the Department of Microbiology and Immunology. She got her undergraduate degree, her bachelor's in veterinary medicine, and then was a practicing vet around the world, really. Africa, Asia, Europe, Australia, Antarctica. And then went on to get her PhD at UC Davis. Got her master's, or your PhD in ecology and your master's in epidemiology. Thanks. She did her postdoc at Penn State at the Center for Infectious Disease Dynamics, and then moved here 18 months ago to MSU. She has a lot of publications and presentations. I just looked through them and was like, wow, this is a lot. And then looked through her honors. She's had a Fulbright. She's had a Centenary Scholarship. She is a David H. Smith Fellowship in Conservation Research. She has that right now, I believe. Or you did, I heard so. That was your postdoc. Okay. Anyways, it was really fun to read through her CV. And again, as I said earlier, a really fascinating work history and a really addition to the rough cut. A little, what should I say? Out of the realm, but a nice interdisciplinary mix up to the rough cut. So thanks very much. Yeah, thanks for being here. I should have found out how I advance these slides. So I just go back and forth with the arrow here. So, yeah. Okay. Good. Okay. So thank you very much for having me. And when Kathy asked me to do this, she said it was a rough cut, and I've taken that literally. So I'm giving slides I've never given before. This is not a polished talk. But the nice thing is I'm going to show some data that's hot off. We're not even hot off the press because we've just submitted some we're about to submit, and some still in analysis stage. That's when I talk about bighorn sheep a bit later. But I really wanted to have a conversation with you all about what disease ecology is, what the role of infectious disease is across the landscape. It's something I haven't really talked about since I've been at MSU. And just to, in the spirit of rough cut, I take complete responsibility for the typo that was in the title. A couple of people have emailed me, they got your title wrong. I fixed it here. But that was my typo, not yours. So firstly, infectious disease ecology, I know that there's few people, I think, on campus really understand what that is. And terms like epidemiology are thrown around, infectious diseases, and people ask me, how is disease ecology different from epidemiology or infectious disease research? A lot of people in our department do infectious disease research. And I guess the way I'd explain it is that epidemiology is the study of risk factors in populations, what makes a person or a population more susceptible to disease, and it tends to be a very static science. It doesn't have a perspective, usually doesn't have a perspective in space and time. Disease ecology is very much about processes that occur over space and time, dynamic processes. And infectious disease research, well, that could mean anything from what a pathogen is doing, how it's binding to a cell, how it's replicating in a cell, how it's spreading through the body of a host. It can mean a plethora of different things. But infectious disease ecology is essentially the study of transmission. And transmission is not like this discrete thing. I mean, I can't take transmission and study it like I could study how a pathogen binds to the receptor of a cell. I And the reason why is because there are so many different processes that have to come together to allow infectious disease transmission. And so here's an example. Here's a pathogen. It's actually something that, this is yellow fever. And as far as I know, yellow fever doesn't infect raccoons, but who knows, maybe it does. But we have pathogens that we can only see in an electron microscope infecting hosts. We have hosts then responding to these pathogens with this incredibly complicated immune response that many people in my department are world experts at. We have innate immunity, we have adaptive immunity. But this play between the pathogen and the host is incredibly important for transmission. Because what happens inside this and how its immune system responds to the pathogen will determine how long does the animal get sick, how long is it sick for, how much pathogen is it producing to transmit, is the pathogen getting to the right tissue to allow transmission. So, for example, influenza has to get to our upper respiratory tract so that we can sneeze and cough it over people for it to be transmitted. And that's one reason why avian influenza is very rarely transmitted from person to person, because it doesn't have quite the apparatus necessary to get into our upper respiratory tract. Or leptospirosis, it's got to get into the kidneys and colonize the kidneys to be excreted in urine to be transmitted. So that complex interplay is really important. And actually how we defend ourselves against pathogens, the immune response can be as important for transmission as the pathogen being there. So for example, to sneeze, we have to be able to have inflammation in our upper respiratory tract that's influencing or irritating nasal passages. And then we can have this explosive action that spreads pathogens across the room. If it was measles, it could be across an entire airplane, depending on the pathogen. Or, those of us who have children, where we know exactly what this is like. They're just petri dishes for infection. And often, if we don't have that host defense, we may not get transmission. So understanding the role of our immune system and what we call this immunopathology. It's actually the damage that our own immune response causes to our system. And a really nice example of immunopathology was done just down the road at Rocky Mountain Lab. Vincent Munster, he is a virologist and a brilliant disease ecologist at Rocky Mountain Labs, and he infected macaques with Middle East respiratory syndrome coronavirus. Has anyone heard of MERS? Okay, so this is a really important and potentially scary pathogen that we think originated from bats, has been transmitted from bats to camels, and has now persisted in camels, we know, for decades across the Middle East and North Africa. And camels then pass it to humans. It's a coronavirus, very similar, very closely related to SARS. Has anyone heard of SARS? The SARS went pandemic. This hasn't gone pandemic, but in Saudi Arabia there have been a couple of thousand cases, and we see these little clusters in hospitals. There was an outbreak in South Korea, I think that was last year. It's very concerning. Anyhow, it causes lower respiratory tract disease, pneumonia, in people who are affected, usually when they're a little sick with something else in a hospital. But Vincent infected these macaques with MERS, and then he treated some of his individuals with steroids so they could not mount immune response. Interestingly, the ones that were treated with steroids did not develop disease. The ones that had no steroids, that had a great immune response, were the ones that showed symptoms of disease. So sometimes it's your immune response that actually causes the disease itself. And so, okay, we've got infection, we've got our immune response, and we've got some mechanism for the pathogen to be released from our body through sneezing or whatever it is. We have to then have exposure amongst individuals to get transmission. And some pathogens are really good, actually, at altering their host behavior to allow exposure. So what's this animal got? Something that kills thousands and thousands, tens of thousands of people around the world every year. Really? Yes. The rabid dog. So rabies is a classic when it affects the brain. It causes the animal, in some cases, to become ferocious, bite the next host, and because the salivary glands are full of the virus, they inject the virus into the next host, and we have transmission. Another pathogen that's pretty interesting in changing of host behavior is toxoplasmosis. It's a protozoal pathogen. Statistically, 20% of you are infected with toxoplasmosis right now. Sorry, it's true. But it's okay. It's all right. As long as you have a good immune system, your immune system has contained it, and you're good. You're okay. So Scott, don't worry. Well, actually, Scott, you might need to worry. But I'll tell you why in a second. So this, interestingly, toxoplasmosis changes the behavior of its host. So the definitive host is a cat. And so if a mouse is infected with toxoplasmosis, the pathogen is going to get its way into a cat. So what does it do? It makes the mouse more risk averse. So is that the right term? Or more likely to take a risk. So a mouse might run across an open field when it otherwise wouldn't if it was not infected, and make itself more vulnerable to be eaten by the predator that is the definitive host of the parasite. Now, interestingly, because so many humans are infected, there have been many studies looking at the same effect in humans. And there was a famous study that showed that motorcycle riders were more likely to be infected with toxoplasmosis than non-motorcycle riders. And people who spend 3 months hiking the Pacific Coast Trail are more likely, they're just kidding. That was a jab at Scott. But yeah, so if you're more inclined to, say, ski the ridge rather than the blue run at Bridgie, you might want to get yourself tested for toxoplasmosis. But don't worry, there's nothing you can do if you have it, so you may as well just ignore what I said. So we've got hosts that have to come together to transmit pathogens. So how those hosts are then distributed across the environment, across the landscape, is going to be a really important factor in whether we can have transmission. Are our hosts all clustered together like this? Well, in some cases, we may be more likely to have transmission if we're all aggregated, as opposed to, say, something that is territorial and nicely, evenly dispersed across the landscape. So that's a really important part of disease ecology, is how are our hosts structured across space to allow transmission. This actually could be great structure, right, for transmission, you think. But in the long term, this may actually be worse than, say, this, because if everyone becomes infected here and then everyone develops an immune response and is then immune to the pathogen, clears the pathogen might actually die out here. So spatial structure over here actually may allow persistence, because we may see pathogen die out here, but we get an animal moves over here, and then we have a little outbreak here, but before it dies out there, they jump over here. We call that metapopulation dynamics. So spatial structure can be very good for persistence. So you're probably, and that should be Wonder Woman, not Superman, sorry, but I meant to go back and try to find Wonder Woman, did not have time. But you're probably thinking, well, disease ecologists are amazing, like they study the immunology, they know about landscape ecology, they know about behavior, they look at these things and these molecular interactions. This is a lot of different scales and disciplines to be able to put together to understand something so basic like transmission. Well, I wish I could say that, but it's actually not the case, because disease ecologists, we are inherently interdisciplinary, because there's no way that any one person can have depth in all of those different fields I just mentioned. So you have to work together across disciplines to understand transmission, to understand how pathogens flow across landscapes. So I'm going to talk about really two different systems here, but I just want to tell you a little bit about what my lab does. So I'm going to talk a little bit about HENDRA virus, and this is a virus within bats, and it is co-evolved with bats, and it spills over from bats to domestic animals and humans. And in all these systems I'm going to talk about, we are looking at those systems from all those perspectives I just mentioned. Not me personally, but in collaboration with big teams of people. So I'm going to talk about HENDRA virus. I'm going to talk about bighorn sheep pneumonia, which in comparison to HENDRA, which is this co-evolved pathogen that lives happily in bats, not causing disease spilling over into domestic animals, we have bighorn sheep pneumonia, which lives happily in domestic sheep, co-evolved with domestic sheep, and spills over into wild animals. And then another project, I'm not going to talk about this today, but just wanted to let you know that we are starting a white nose syndrome project, where another big collaborative team, white nose syndrome is a fungal pathogen introduced from Europe into American bats. on the east coast of America, and it's slowly sweeping across the west and causing local extirpations, many bat species being listed as a result of this fungal infection. It's devastating to bat populations. And we're trying to understand the susceptibility of the western bat populations, so managers can have information to deal with this as it arrives here. It'll probably arrive in Montana in the next few years. So these three systems all have in common that we're trying to understand the flow of pathogen across landscapes, between species, and we're particularly interested in the conservation and management of the wildlife species involved in the disease. First, I'll start with HENDRA virus. And before I tell you really anything about the virus, I'm going to tell you a little bit about the hosts of the virus. So I'm Australian, as you probably guessed, and I didn't actually start studying HENDRA virus until I moved to America. It was all American-based research, although I have a lot of collaborators in Australia. But to understand the ecology of HENDRA virus, we need to understand the ecology of the host. We have 4 fruit bat species in Australia, and these are not the little bats you find in Montana. I mean, these are like a kilogram body size like this, a meter wingspan. And they're all nectivorous. They're all primarily nectivorous. And that means that they have to migrate. So the ecology of Australian forests is very different from the ecology of American forests or North American forests. So in Australia, we have eucalypts. Our forests are dominated by eucalypts, and they're flowering. We call them flowering gum trees. And flowering occurs in pulses across the landscape. So we'll have like a big pulse or a big bang nectar pulse here, and then it will disappear after a few weeks. And then we'll have nectar over here across the landscape. And then that will die away, and there'll be nectar here. And so these bats have evolved to constantly migrate to find food. And so most species we would actually call nomadic, not migratory. And so they have to constantly be on the move. But what we now see in Australia, if you go to any major city on the east coast of Australia, or even any big town, and you wait until it's just on dusk, it's just dark, and you look up in the sky, you will see fruit bats, or we call them flying foxes. And some places like Brisbane, the sky is literally full of these big black objects flying around. And this is Sydney here, right in front of, in the Botanic Gardens, in front of some of the tallest buildings in one of the, well, the most heavily populated city in Australia with fruit bats. And that is a totally new phenomenon. So what's happened is bats, instead of migrating constantly across the landscape for food, and now moving into towns and cities and staying completely sedentary. So we're now seeing a majority of the bat population in these urban populations and not moving. Before 1981, there was never a record of a sedentary flying fox or fruit bat in Australia. So this is a totally new phenomenon. And this is just an explanation, more a bit of the ecology here, that we see nomadic behavior in bats for nectar and pollen, which has very high productivity, very high energy, and high protein, nutrient content. Seasonally, they would feed on fruit if there was a shortage of nectar. So they'd come into rainforest areas and feed on fruit. And then, and this is historically, if there was no nectar available, there was a big food shortage, they'd then feed on these starvation avoidance, low quality, low density foods. This is survival food. What we're now seeing with bats in Australia is that stopped migrating, moved into urban areas. They're eating fruit in people's backyards or whatever they can find in the city, which is essentially much, especially at certain times of the year, starvation avoidance food. It's like junk food. So basically moving into the cities and eating McDonald's for bats.
What's that done with numbers?
Well, there's been decline in all of 4 species. 2 of them are listed as vulnerable. And interestingly, there isn't a lot of information on the ecology of bats because I think, well, partly I think Australia's only got 20 million or 22 million people now. So there aren't that many ecologists in Australia. I mean, we're talking about like the population of the lower LA basin filling the whole continent of Australia. And then I think as well, bats are considered like vermin. They're, I'm trying to think, like, would there be like a white-tailed deer I mean, there's not really a great interest in them. They're considered to be dirty and smelly, because especially they come into cities now in tens of thousands and roost in people's backyards. And they're very noisy, they smell, they poop on everyone's laundry.
What would be the lifespan of a bat that was urban versus non-urban?
We have no idea of that. And that will be pretty difficult for us to tell, but we're starting a whole suite of studies to try to understand the health of urban bats versus migratory bats. But in captivity, bats have lived late into their 20s. I think there was a captive bat that was 28 years old. In the wild, we suspect their average lifespan is about 10 years. So they're very long-lived. They're slow breeders. They're slow to sexually mature. They have one pup a year. So these are, even though they're very small mammals, they have anything but the strategy, the life history strategy of a small mammal. They have more of a life history strategy you'd expect of a large mammal, which makes them very vulnerable because they can't just, they can't suddenly reproduce like a mouse and make up for these stresses. And there's a lot of stresses on these bats, and I wasn't going to talk about this, but since Douglas is in the audience, and I know he's interested in climate change, I should mention it. But one of the big stresses is that we're having these heat events in Australia now, that we're having them almost yearly, where we're seeing 48 degrees Celsius temperature, which I think is 118 degrees Fahrenheit. We'll see a couple of days of this extreme heat around Sydney, Brisbane. And during those days, we'll see thousands of bats die. I mean, they just literally fall off the trees, and there'll be piles of dead bats under the trees. And then we've also seen bats go into distributions that they were never found before. So historically, bats would only come into these southern regions when there was a severe food shortage, or in the western regions. So they're pretty much defined to this northern extent of Australia. We now have permanent cancer flying foxes here and in Adelaide, which we never used to have flying foxes. And these areas can be very cold. So there's potential cold stress as well. So we have heat stress and cold stress. And by the way, those red dots there are where HENDRA virus spillover events have occurred from bats into horses. And so one project we have, we're just starting in this lab. This is my PhD student, Maureen. She's going to be looking at the energetics of bat migration and tying that in with viral excretion. And so just very briefly, what we think is happening is that there's been like some sort of a threshold change in the availability of nectar. So if you think about forests across eastern Australia being like this source of energy, and I mean, it's really about distribution of energy. And 80% of those forests are now cleared, and there's continuing, ongoing clearing, especially in areas that are really important for bats in winter. And so we have a migratory species that can no longer always find a source of nectar when it moves across the landscape. And so we think this is basically an energetic decision. Bats, I don't think the bat flies along and says, is nectar available? But basically, if there's nectar available, yes, well, it's probably going to try to find nectar because nectar is very high in energy and nutrients, as I said. But is it cost effective? Once there's less nectar and once it's more variable, harder to find, climate change, again, is a big issue with production of nectar, so we think it's becoming less reliable in time and more synchronized. If it's not cost effective to move to find nectar, then maybe we don't migrate. Maybe we actually should become residents and go into cities. And then we think then they're subject to a whole bunch of stresses, particularly nutritional stress. And we suspect that that's that nutritional stress that's then linked to HENDRA virus shedding and spill over to horses. And we've done some big field studies that I'm not going to talk about. I feel like I've talked about them too much in the past, but suggest a link between nutritional stress and virus. So essentially we've got forests being converted and fragmented, changing the spatial distribution of energy across the landscape. And that's leading for bats to come into urban areas, urban bat habituation. And we think also an energetic issue is causing increased bat excretion of virus through nutritional stress and other stresses. And then we're seeing spillover of viruses into horses and humans. And just to show you that these new urban populations are really important, Here we have these dots here are locations where bats will roost. So they will be full of bats when there's nectar around and empty when there's no nectar. These red dots here are the urban bat populations. And this area, this is Brisbane here, and this is incredibly urbanized around here. So these are urban bat populations in this region. These are sedentary, they're always there now. And these triangles are HENDRA virus spillover events. And we've done some statistical analyses, not yet submitted for publication, showing that there's a statistical correlation between urban bats and HENDRA virus events in horses. So what happens when we have a HENDRA event in horse? Well, it's a huge deal. HENDRA virus is about 70 to 80% fatality in horses and humans. So it's these kinds of pictures we see when a horse is infected. HENDRA virus has completely changed the veterinary industry in Australia. So there's been now, I think, 54 spillover events, but over 100 horses infected. But horses will present with basically anything like from colic to neurological disease to a fever. So a veterinarian can't predict that a sick horse is going to be infected or not infected. Vets now have to treat any sick horse with this kind of protective gear. There's a whole big argument about vaccination. I do not have time to go into it, but it's fascinating and more complicated than the disease, I think. And it's increasing we're seeing an increased incidence of HENDRA over time. So our first outbreaks were in the mid-90s, and we really just saw it sporadically until about 2006. And now we're seeing HENDRA virus events every year. We tend to see big clusters of HENDRA virus spillover to horses. And this time scale is all correlating with the time scale of bats moving into those peri-urban, urban areas close to where we keep horses in Australia.
How does it translate from the horse to the bat?
So we think bats, when they're roosting,
Bats to horse.
Yeah, so bats feed in horse paddocks, or these urban bats. So this is a really big difference between migratory bats. So migratory bat feeds in a forest, native forest. These urban bats feed in trees like fig trees planted in horse paddocks. So they have this extra opportunity for contact with horses. And we think what happens is the bats are defecating, urinating in the paddock, and horses will come and graze infected grass from under those trees where bats are roosting, or not roosting, feeding.
So if they're in the urban area, there's got to be potential contact with way more than horses. Is it just that horses are particularly susceptible?
Yeah, that's a really good question, and we don't really know. We know dogs now are susceptible. In fact, I would credit all of my funding to a dog getting infected because, I mean, people care about horses, especially in Australia, but when a dog gets infected, this poor dog's picture was on the front of every newspaper in Australia with its eyes kind of like this. So probably many different domestic animals are susceptible because many of these bat viruses seem to have a very broad host range. And I think the horses, we see it in horses because horses have that contact opportunity, that they're sniffing around on the ground around bat feces and bat urine when like a cat or a dog just doesn't have that same kind of, they're not out under trees where bats are feeding. We've never seen a human case directly from a bat, and that's interesting too. We think, and I'm going to get into this in a sec, we think bats have a very, very low viral load. I'm going to get back into the immune system in a sec and explain why that is, but we think that might be why humans don't get it directly from bats. So we now know that bats, when they excrete virus, they tend to do it in big pulses. So we'll go out and we'll look. We looked for HENDRA virus for years. We could not find it. Eventually we had to get more sensitive techniques, but we still will go out, we'll find very little, or we don't find it for months after months and months. And then all of a sudden there'll be like 70% of the bats excreting virus. We call this like an epidemic, a big pulse of virus excretion. And my lab we're all about the pulses. We're like, why are we seeing these pulses? We do think there's a nutritional component. I'm not going to have time to go into that, but I just want to say, when bats excrete a pulse of HENDRA virus, these are different viruses that people are looking for, and you can see nothing, 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 nothing. Big pulse. HENDRA virus is the green. That's also excreting a whole bunch of other viruses. These are just paramyxoviruses. Paramyxoviruses, we know them well in humans. Measles, distemper, Newcastle disease, really important public health and veterinary diseases. These are a whole bunch of newly discovered paramyxoviruses all being excreted at the same time as HENDRA. And we're now seeing this in bats internationally. We're seeing it in Asia, we're seeing it in Africa, that bats are excreting a diverse range of diseases. And we know that a lot of them may be zoonotic. You know what zoonotic means? Zoonotic means a pathogen that's capable of being transmitted from an animal to a human. So humans are susceptible. And there have been some really prominent ones recently. Ebola, SARS. Here we have, this was actually published before the West African Ebola outbreak, but we have, these are just prominent zoonotic diseases, but the bat viruses dominate. We've got Ebola, we've got HENDRA, Nipah virus, almost identical to HENDRA, that infects humans in Bangladesh every year. That was the basis for the movie Contagion. Anyone see that? A really scary virus, because that does go from bats to humans, and we see stuttering chains of human-to-human transmission, 70% fatality rate. And this is one that WHO is holding its breath over. Because if there's just the right mutations for that to be transmitted effectively, we will be in Contagion, the movie. So SARS, MERS, I talked about MERS recently. And there's a whole suite of other ones that just occasionally spill over into humans, and they haven't made the limelight yet.
So are these all just seasonal? Like, do they always happen in the same, month of the year?
That's a really good question. That's exactly what we're trying to do right now. So I have a postdoc in my lab and analyzing 12,000 samples of bat urine. Actually, he's not analyzing the bat urine. He's analyzing the results of the samples. So, and exactly what we're trying to do is trying to see, is there a seasonality? And there was some, the really rough cut analysis showed probably an annual pulse in winter when bats have the least food available. But now David is getting into all these nice time series analysis techniques and showing, actually, we're probably seeing little pulses every 3 months that we're not detecting with the appropriate statistics. So, yeah, that's a big question right now. And I'll show a bit of that. Yeah.
What's the percentage of horse cases that actually resolve in transmission?
Very few, very few. It's actually pretty hard to get it as a person from a horse. And there have been 7 people infected with HENDRA, and 4 have died. And everyone that was infected either had like they were doing a necropsy on a horse, and they asked for the armpits and basically like blood and everything else, as you do when you do a necropsy on a horse in the field. Or one veterinarian was infected when he did an endoscopy, so basically sticking a tube down the trachea to look at the airways, and he basically, he would have got full face full of virus-laden respiratory secretions.
And they're not transmitted from human to human?
No human-to-human transmission. And we don't know whether it's possible or whether it's just that once humans have symptoms, they're put into a biosecure isolation ward. So HENDRA has the same biosecurity rating as Ebola or Marburg. It's a BSL-4 pathogen. You can't actually deal with it without being in a spacesuit in a special containment laboratory. There's only a few around the world that you can do that in. And it's like a container within a container within a container, and then even in that container you have your own air supply that's external. It's, people are very cautious. So with the bats, we think that there's probably something very special about bats and why we're seeing all these diseases emerge from bats. And this paper, I'm just putting this up because this was out last week in PNAS. This is led by Michelle Baker, a colleague of mine. And I know they've been working on this for many years, but this just shows bats have some very unusual immune adaptations that may allow them to host a suite of different viruses without getting sick themselves. So bats don't get sick from HENDRA, Nipah, Ebola, Marburg, coronaviruses. They're healthy, as far as we can tell. There's no pathology or disease associated. And we now think bats have a special mechanism to allow that, which is related to their ability to fly. Yeah, interesting. So bats, I mean, they're the only mammal that can fly, which is quite fascinating in itself, isn't it? And they have, I mean, quite different adaptation to flight than birds. So when a bird flies, it increases its metabolic rate twofold. When a bat flies, it has to increase its metabolic rate 16-fold. And there's a dire consequence to having a high metabolic rate, and that's oxidative stress, production of free radicals that damages DNA. And now a number of groups have sequenced the genome of bats and found that there are specific regions on the genome that are associated with trying to prevent oxidative stress, the DNA damage, that also are involved with immunity. So for example, this paper, they show that bats have this interferon. Has anyone heard of what an interferon is? It's an immune molecule. It's part of our innate immune system. If we're infected with something, we produce interferon, a big pulse of it, and that helps us to combat the pathogen. Bats have just this high baseline of interferon all the time. And that, by bathing cells in interferon, they've been able to show that can reduce DNA damage from oxidative stress. So we also had a paper out a couple of years ago, it's called now the flight as fever hypothesis. Bats fly and have a higher body temperature, and high body temperature fever is one way that we also reduce pathogen replication. We suppress pathogens when we're infected. In places like Australia, people don't treat fevers like they do here. You know, my kids, right, I always have to treat them when they're over 101.5. In Australia, you don't treat kids until they're over 104, just because you want to let the fever play its role in taking care of the pathogen. So perhaps bats, by flying and having a high body temperature day in, every night, that perhaps that's another mechanism that's at least driving selection for pathogens that have a low rate of replication. So just, I want to go quickly, because I do want to say something about bighorn sheep, and I know I'm going over with HENDRA, but this data set, we're analyzing a data set of 4 years of looking at urine from underneath bat colonies across the east coast of Australia and looking for trends in pulses. And this is my postdoc, David. I want to credit him. He's doing some really cool time series analyses that I wanted to show, but I can't really explain all of it, so I'm not going to. But so that, you know, I hold your breath for that. We'll present that soon. But this is just, I think, a nice overview of what our lab's doing on HENDRA virus. That we're interested in the migration of bats, urban habituation of bats, in relation to the dynamics of the virus and their foraging ecology. We're interested in how land use change and human population growth is driving this whole dynamic here. And we also, and I'm not talking about this today, but we also are looking at the spillover. How does pathogen get from the bat to the horse? What are all the various things that have to happen for a pathogen to spill from one species into another? And then we are collaborating with social scientists to try to understand all the human dimensions of horse management and behavior, peri-urban land use, the big swing towards culling bats that's happening in Australia, these urban bats, and how that then feeds back into stressing bats and causing more virus spillover. So it's a big integrated system, and that's an overview of what we're trying to do. I think I've got 10 minutes to talk about sheep. So bighorn sheep in this region pretty much dominated or covered this landscape. There are some estimates that there were probably 2 million bighorn sheep before settlers came. Now there's less than 70,000 bighorn sheep. And all the work I'm going to talk about today in relation to what's driving bighorn sheep population decline, I want to acknowledge this person, Frances Cassier. She spent 20 years in the field collecting data on bighorn sheep pneumonia, and she works 100-hour weeks to collect the data that we use and collaborate on the field projects that we do. So the area that we're trying to understand bighorn sheep pneumonia. So let me go backwards a little there. We've seen these massive declines of bighorn sheep, and we know hunting was a part of it, and it certainly isn't a part of it now. Hunting's very restricted, bighorn sheep. pathogens, pneumonia has been a major source of the original decline and is the reason why we can't recover these populations. And I'm a little out of order here, but this is the system that we're working in, which is right on the edge of Idaho, Washington, and Oregon. And we have 16 interconnected populations that we've been looking at data for over 20 years from 500 animals that are radiocollared. This is what it looks like there. It's beautiful. It's wild. This is the Snake River here, and the sheep live up on these cliffs along the side of the Snake River. There's about 800 bighorn sheep there. They were extirpated from there by the beginning of last century and then totally reintroduced. And so bighorn sheep pneumonia, what we understand is that it's caused by a pathogen called Mycoplasma ovipneumonia. Domestic sheep co-evolved with it. If you go into any domestic sheep population that's probably over 50 animals, you'll find it. You'll find it out here in your backyard. And it causes no disease in domestic sheep. Well, that's the theory. We actually think that theory is wrong, but generally no disease in domestic sheep. You go into a population of domestic sheep and you'll find dozens of strains of this bacteria. So it's very diverse. That's what you would expect in a co-evolved chronic pathogen. But when the sheep give mycoplasma to bighorn sheep, we see these big die-offs, or so we thought. That's what we really thought. This has been the paradigm of bighorn sheep pneumonia. This is what you read in the Chronicle, right, or the Billings Gazette, is that there was a population of sheep and now 50% of them or 90% of them are dead. They have pneumonia. That's been the paradigm. And mycoplasma causes disease. It binds to mechanisms in our respiratory tract. There's this cilia that allows us to clear pathogens. It destroys the cilia. It allows anything that's in the nose just to fall into the lungs. And they die of it, basically a bacterial soup, pneumonia caused by a whole bunch of different bacteria. So in Hell's Canyon in 1995, Mycoplasma ovipneumonia was introduced into a population. These populations have been growing exponentially, the dotted here. We saw disease introduction and then a major crash. And then since then, we've seen populations increase and decrease, some kind of be stagnant, some decline. One actually has now gone locally extinct. But generally, they don't do very well once this pathogen is introduced, and they don't clear it. They tend to remain infected for a long period of time. This is a connectivity analysis that we did with Brett Dixon, showing that all of those, this is that same map, showing that all those populations are highly connected. Bighorn sheep can interact across that big landscape, and hence we saw that 1995 outbreak, that big dip, started here, but then it spread quickly throughout the whole metapopulation. And now we have every population except one infected, and we saw Sheep Mountain blink out recently. So we've witnessed an extinction event. We have a whole bunch of people working at all different angles. We work together closely. And I think one of the major findings we've had is really defining the signature of this disease. And it's not these big die-offs of adults that you see in the newspaper. Yeah, we do see the big die-offs of adults, but after those populations have gone through the die-out, the adults actually survive really well. Here's healthy, diseased adults of different ages. And statistically, we can't distinguish survival of adults in diseased populations and adults in healthy populations. So what's causing them to be depressed? Why are they depressed or declining or going extinct? Because of lambs. So adults develop immunity to the disease effects of pneumonia. They can be infected, but they're not dying of it. But every year they're giving it to the lambs. And this is the burden of disease over the 20 years. The burden of disease, the animals that are dying of pneumonia are the lambs every year. And in a healthy population, we'll get 80% recruitment of lambs. In a diseased population, we'll see less than 20% recruitment of lambs. And they're not dying slowly, just over some period of time. They're actually dying in a pulse. So here's the healthy, this is the daily hazard of mortality. In a healthy population, of course, you can perhaps be more susceptible to predation and so on early on. In a pneumonic population, the lambs are dying between 4 and 14 weeks. We see all the mortality within this period of time. And it makes sense because mycoplasma has to colonize those cilia. All those other bacteria have to slowly fall in. We get this slow development of pneumonia and then really sick lambs and they die. So I'm going to skip that one. But this is recruitment, the survival of offspring, so they recruited into the population. Recruitment in a healthy population should be around where this purple line is, or actually even higher. This, I think, is in biased low. Recruitment in these populations has remained, here we are 18 years after the disease invaded, recruitment has remained low. And this is what's suppressing these populations over the long term. And some simulation modeling that Kezi and Manlife has done to the PhD student just finishing up, and a whole bunch of things about to be submitted here. This one's in review. But just look at this gray area here. She has simulated, given the data we have from Hell's Canyon, if we then take it out to 30, 40, 50 years, we expect this very slow decline. And the only reason we can even see this is because we have so many animals radio-collared, and we're looking for lambs from radio-collared ewes every summer. And if you were in a population that's not being monitored like that, you'd miss it, because this is, I mean, if you look at over 10 years, you won't really even see this. This is what we call like an extinction debt. This is something where these populations are declining so slowly. We're not expecting extinction for 30 to 50 years. But if you think about it in a long time scale, this is a dire issue for bighorn sheep. So we asked, well, how is this pathogen persisting for decades? How is it passing around from individual to individual, or do we have carriers? Who are the carriers if they're there? Are all lambs dying equally? So we started some studies and some very intensive field studies in 3 populations. And the reason we're able to do it, and I'm going to talk a bit about Lost Dean for just a couple of minutes, is because this man, Dick Coggins, he was a manager in the Lost Dean population for 30 years. And he's just such a wonderful human being, but he would go out to in a blizzard like this with his notepad, and he would write extensive notes about which sheep had a runny nose and where the rams were on the hillside this afternoon. And, anyhow, his presence there and his feeding allowed the sheep to be habituated. And so we have this population that, rather than having to spend $10,000 per helicopter capture, we can actually walk right up to. And these animals, as you see, they're right next to Vic here. She's my favorite in the population, number 9. And she's infected with mycoplasma. As you can see, her snotty nose. And I swabbed that snot. It was full of mycoplasma. And but she's so sweet. Like, she'll come right up to you and rub herself against you. I mean, you could stand there for like an hour just scratching her back, and then an hour later tickle the tick off you. But she's very sweet. She's 18 years old. But what we found here is that the population was really divided into carriers. We had animals that were always carrying mycoplasma, these red dots. We had animals that were always negative, these blue dots. And then we had some animals that were shifting status from positive to negative. And interestingly, we found those animals that were positive, these chronic carriers, were more likely to be old. They were the older animals. They're like number 9, that old 18-year-old who, they were these guys. But also lambs surviving. The lambs that survived, most lambs die each year, but the lambs surviving were all positive. And we're suspecting from just, I'm talking about like N equals 3, we're suspecting that as they move into prime age, they're clearing the pathogen, and that as they're getting older, they're more likely to become infected. But this is really important because it gives us a potential management tool for thinking about perhaps removing carriers from populations and allowing recovery.
So when you, those kind of data, are they based on visuals, like a runny nose, or are you actually taking blood samples and looking for antibodies?
So we take a nasal swab of each animal. We've done, most of the population was sampled at least 3 times, some 4 or 5.
Are you meaning that they're no longer visually symptomatic, or
no, because
cleared the pathogen?
Because actually many of the infected animals look beautiful and healthy, and have not got a runny nose at all. You cannot tell visually an infected. So we're only telling from doing PCR on nasal swabs. So we stick a swab in the back of the nose, we do PCR and look for mycoplasma, epidemiological.
They couldn't be further back in the system. I mean, what I'm going to this question is, as an animal matures and the immune system becomes more robust, would it suppress it down to such a small, low level that or in places where it's difficult to sample, but that they're there, and as they get older and the immune system starts to rust away, then it becomes symptomatic.
It's a very good question, and to actually answer that, my collaborators have taken some of those intermittent animals, the negative animals and positive animals, into captivity. The only way we could answer that is by putting those negative animals with known susceptible animals that have never been exposed before and seeing whether they can transmit, and then repeat sampling and sampling. Those animals have stayed negative, they haven't transmitted. But it's something that's really a good question, especially for those animals that we see sometimes positive, sometimes negative. Is that because there's spatial heterogeneity in where the pathogen lives in the nasal cavities? If we happen to stick the swab in this way, but the pathogen was that way, are we missing it? There are questions that are so difficult to answer, and really we have to have a captive flock that we can, or a captive herd we can monitor over time. Yeah, but there's so many uncertainties like that in disease ecology.
So what's your opinion then on that experimental shoot-off of that herd, where the pneumonia has, they've never been able to, the herd has stagnated and they couldn't grow. And the hypothesis, as I read it, was that this pneumonia was persistent, and as passing lambs, recruitment was nothing, and so they're going in and shooting off the entire herd and then transplanting fresh.
I'm going to show you some data in a second that may actually bring that into question. 5 years ago, I would have said that was crazy. I can't tell you that's crazy anymore. But I'm hoping that we can start to figure out which animals are carriers, and there may be some management options for us to actually let those populations recover. We're trying all sorts of things with models, with experiments, field experiments to see whether there is a way we can do it. But at the moment, I can't tell you that's a crazy thing. I think this disease is so intractable.
Sorry, I have a quick question. Are all the herds that have mycoplasma doing badly? Or are some herds that have carriers and that have mycoplasma that are doing just fine?
No, I wouldn't say there are herds that are doing just fine. We see a really big spectrum in outcomes in infected herds, which is it's the major question of our research project, is why are some herds able to plod along with a stagnant population growth? Why are some, like Sheep Mountain, going extinct? We do see some populations that are growing at a very low rate. We don't see any population that's infected doing what they should be doing in Hell's Canyon, which is growing exponentially. And then they should be able to have a lot more sheep there. There are a couple of herds that have been real outliers, like Missouri Breaks. It's a real example where there are infected animals and they're doing okay. And one thing I think is really important is the spatial scale that you look at the problem. And Kezia's work was really nice, showing that the scale of disease can be very localized. So it could be localized to a U group. So you can have 3 or 4 U groups in a population and one doing really poorly, and the lambs all dying in that group. But if you come and do a population census, you're basically averaging over this multiple populations, and that population may look like it's actually doing okay because the other 3 U groups are good, but there's no disease or transmission there. So that's a big problem. And I know a lot of people in this region have said, oh, well, the sheep here are doing really well. But when they've actually gone in and started looking at the appropriate spatial scale, like, well, actually, this group's really not doing well. This group's doing great, but they're not infected. But why some populations do poorly? And we think actually strain is a big part of that too. And this, I've only got a couple of minutes, but I want to quickly tell you why we were studying these populations. We had an event happen that I think has made me feel much more dire about bighorn, I think the long-term viability of bighorn sheep, and that is we had an infected population of ewes that were chronically carrying mycoplasma. They'd been infected for over a decade. The ewes were surviving really well. Their lambs are dying every year, as we'd expect. We then had another strain of mycoplasma. It was actually a goat strain be introduced to this population while we happened to be doing this, or Kezia was doing this intensive observation period. So she was looking at those sheep many times a day. And so that was in 2004. Just to compare, this is a survival curve. 2013, all the ewes survived. 2014, when we had this introduction, we lost 30% of the ewes. Both years we lost all the lambs. You lose lambs, whatever. But 30% of ewes, I mean, that's like what we would see in a naive population that first has an outbreak of pneumonia. And so we've done a whole bunch of different analysis and strain typing and sequencing and found that It was basically a goat strain of mycoplasma that came in. And now we've detected a whole bunch of different strains in Hell's Canyon. But the reason why I say this makes me feel very concerned is because if you go into it, as I said at the beginning, if you go into it just a domestic sheep flock, a backyard domestic sheep flock, you will find dozens of strains of mycoplasma. And if sheep don't have immunity across these different strains, we could have this basically infinite source of new pathogens for bighorn sheep. That even once they're in this infected stage where the lambs aren't dying, the adults are surviving and they're just teetering along, they're still at risk of a pneumonia outbreak if they have contact with sheep or goats. And this group of sheep, the management effort to stop any contact with domestic sheep or goats was enormous. I mean, the people were going to the owners. I mean, there were so many efforts there that it failed. And so this makes me worried about the ability of bighorn sheep to survive on our landscapes if there's continual contact with domestics. I was going to talk about immunity, but I'm out of time. Oh, there you go. That was the only problem with a max to PC problem we've had is the last slide. So I think that's actually pretty good. But I think the big picture, the future of bighorn sheep, vaccines are very unlikely to help, especially with all these different strains. Separation strategies are very difficult. We have to be really careful translocating. And I think the best hope for bighorn sheep is we need big landscapes where there's no contact with domestics. That's the best hope for these species to survive. Wonderful group of collaborators I have here. Some lovely funders. And I probably have like 30 seconds for questions, if that, maybe. Do I have time for a question, or are we?
I think so. I think if some people need to go, and then you do at 12, that's great. Just stay an extra 5 minutes or so, too, after 12. We'll probably ask them after a while.
Yeah.
So are there, in state of Montana, I'm not talking about your study populations over in Hell's Canyon, but in Montana, are there any islands of disease-free sheep that can be used to transplant back in?
There are, yeah, there are definitely disease-free populations here. And the state agency here, I think, is actually really sensible about, like, they're very aware of this issue and very cautious about these sheep, maybe more recently than historically, but there is a real move now to only translocate from animals that are disease-free. And we can tell through serology, like looking at antibodies in serum, whether they've been exposed or not. So as long as we sample enough animals, we can tell which ones are disease-free. But there's no point putting a population anywhere where they're just going to have contact with domestics. So that's the big problem, is like, where can we actually put sheep where they cannot have contact with domestics? Like, historically, we've had all these boundaries, like it was 6 miles and it was 20 miles, and now we're thinking more like 100 miles or more from domestic sheep.
What's the ecology of mycoplasma once it hits the soil? I mean, so for example, they're
dead.
They're dead.
Doesn't have a cell wall. Dead. Yeah. It doesn't survive outside a host at all. I mean, we actually have seen it. We've seen it transmitted from pen to pen across like distances from meter Scott through respiratory secretions, but that pretty much has to be directly transmitted. It doesn't survive at all in the environment. Great, thanks.